All right, we're ready to start. Good afternoon, good morning, good evening to those of you joining us from all over the world. My name is Lisa Osipenko. I head Consilium Scientific, and today we're very, very pleased to welcome Professor Frank Harrell. He's a professor of biostatistics from University of Vanderbilt Medical School. And you can read Frank's bio on our website, but one thing I would like to say about Frank is based on my experience teaching technical subjects, I wish I could be anywhere close to Frank in terms of teaching skills and conveying difficult concepts, difficult methods to students and to wider audiences. So you're in for an excellent treat. And today, Frank will be talking about clinical trials design and why many clinical trials don't pan out as they are planned to be. So Frank, please, the floor is yours. Go ahead.
Lisa, thank you for that nice introduction, and thank you for inviting me to give this presentation. I've been working in clinical trials for many years, and as a biostatistician, have a lot of methods that I like, and some of them I want to show to you today. I'm also an advisor to the Center for Drug Evaluation and Research at FDA, and working in the rare disease arena and trying to learn what are the issues that FDA faces all the time, and that informs a lot of what I do. I want to jump right into experimental design, and really the heart of experimental design is that physicists know how to do experiments pretty rigorously, and they know how to get reliable and reproducible results. They don't tend to do sample size calculations. They tend to experiment until they have an answer. And some people think that's really bad. They think you can get anything you want if you experiment till you have an answer, and that's really not the case. And physicists have shown that over and over. So why is it that medical research, and especially drug development, is one of the very few fields that operates in a completely different mode? So can we act like a physicist and do really sequential learning, sequential experimentation? So what we find is in biomedical research, sequential experimentation is almost never used. And it really offers a lot. Sometimes it even offers more complex things, such as adaptive trials. So why do you not just collect data until you have sufficient evidence in one direction? The direction could be benefit, could be harm, could be non-inferiority, whatever you're interested in, and keep going until you run out of time, money, or patience. From a scientific standpoint, there's absolutely nothing wrong with that philosophy. But we tend to engage in voodoo when we do sample size calculations, and any statistician will tell you that sample size calculation is where we feel like we're doing our most embarrassing work. It's a big game that some people call the sample size samba, and we would like to get away from that. And so, excuse me, going the wrong way. So one of the things that's keeping us from doing sequential experimentation and learning more rapidly and learning as we go is traditional statistics, which is usually called frequentist statistics. It's sometimes called sampling-based statistics. It has a big impact. And one of the things to me that is the most lamentable in my career in statistics is how much we are driven by history and tradition. And we can't seem to break out of that and to use more modern methods. Another contributing factor is fixed budgets. So budgets seem to be approved for doing a certain sample size and enrolling so many patients in a trial. And our National Institute of Health in the US really budgets fixed studies for the most part. They don't do portfolio budgeting. So a pharmaceutical company can do portfolio budgeting, and they have a lot of flexibility, especially using a lot of the methods I'm talking about today in phase 2 and phase 1 studies. But budgeting has a lot to do with our limitations. But wouldn't it be more logical to direct more resources to promising studies, even if you get to the planned sample size, and direct resources away from unpromising studies based on things like futility analysis? Now, this gets at the heart of the different schools of thought in statistics. And if you've studied how clinical trials are taught traditionally, they use a concept called alpha spending, and it's really based on a very rigid design setup. And when you're using that sort of traditional setup, it puts a lot of limitations on the design, and it also turns out to be quite conservative with regard to sequential experimentation. So I tried to put together this simplest way of distinguishing Bayesian statistics from traditional statistics, where the first of each pair of bullet points is the traditional statistics, frequentist statistics, and the second bullet point in the pair is the Bayesian approach. So would you rather know the probability of a positive result, the probability of getting a positive result when there is nothing? In other words, when there's no treatment effect. Or would you rather know the probability that a positive result turns out to be nothing? Those statements look a little similar. They could not be more different from a standpoint of evidence generation and probability. So the Bayesian approach is interested in the probability of making a mistake. In other words, a probability that the drug really doesn't work when you say it does work. And that doesn't really come into play in the traditional role. So in the traditional frequentist approach, you calculate p-values and confidence intervals, and a p-value is measuring how surprising your data are if the treatment has no effect at all. It doesn't hurt, it doesn't help, has nothing. So how surprising are the data if you were to make the assumption that the treatment doesn't do anything? Whereas the Bayesian approach will get you the probability of all possible levels of efficacy. It gets you evidence in favor of something, evidence against things, not just evidence against a null hypothesis. So these are two ways to distinguish the two. And how you design studies, it has a lot to do with whether you're using a traditional frequentist approach or not. So if you like the first approach, you like the status quo, and a p-value is used throughout where you calculate the probability that the data are more impressive than our data if the treatment is completely ignorable. If you like the second approach, then you have Bayesian tendencies, so watch out. And you like posterior probability. So with a posterior probability, you're calculating the probability that some unknown effect is greater than some constant C, conditional on or given the data and the prior information if there is any. And so examples of things you calculate very directly with the Bayesian approach, the probability that there's any blood pressure reduction, the probability that the reduction is clinically relevant, for example, greater than 5 millimeters of mercury, the probability that two drugs are similar. In other words, the change between treatment arms in the systolic blood pressure is on average between -3 and 3. So those would be Bayesian probability statements. And you can see already that these are quite direct in their actionability and interpretation. Whereas here, if you say that a p-value is small, well, that's giving you some ammunition that your assumption of the null hypothesis is not a good assumption. But that's a much harder thing to action, whereas these kind of probabilities lead right into decision theory and optimum decision making. So in power and sample size issues are really related to the statistical school of thought that you happen to be in. And this leads into a discussion of what are the most common outcomes of clinical trials. Well, insufficient patient recruitment is the #1 outcome of a clinical trial, unfortunately. And then the second most common outcome is an equivocal result. And this is really a sad thing because at the planned sample size, the results are, quote, not significant, but the uncertainty intervals are so wide that you haven't learned anything. So the number of clinical trials that are published on a daily basis in which the confidence interval is so wide that you don't know anything after the trial was done that you did not already know before the trial was done and the resources were invested. It's an amazing high frequency occurrence. So you've learned how to spend money and time and resources, but the equivocal result doesn't go any further than that. And so one of the causes of an equivocal result is an over-optimistic power calculation. it is still such an incredible game that people play by choosing a clinically relevant effect size and then maybe calculating the budget needed to support that. And then all of a sudden, the effect size to detect magically doubles. And so people are designing studies where the effect to detect is more than the minimal clinically important difference, or MCID. And what happens is if the effect is only clinically relevant, you're likely to miss it because you powered the study for a double clinical relevance, and you may only get a single magnitude of clinical relevance. Fixed sample sizes are a big cause. So if you get to the end and you say a little bit more evidence would push us over the edge where we could say that we know the treatment works, well, in the frequentist world, you've already spent alpha, and that really holds you back. And so this loses power and results in a huge number of equivocal studies. And when you take multiple looks at the data, the fact that people think you need to preserve alpha is making the assessment, sequential assessment, overly conservative. Another major cause of equivocal results is people are still using insensitive outcome measures. I've seen studies of rare diseases where the most patients that could be obtained was in the range of 40 patients. And the sponsor made the outcome into a dichotomous variable. So everybody should know that's a minimum information, minimum power outcome measure. And you just can't afford to use an outcome measure that's binary in a rare disease situation. It's just a recipe for disaster. So another common outcome is you have a decent sample size, but you have little evidence that the treatment works. Well, that's not in and of itself a bad thing, but it's actually, most of the time, is actually tragic. Why do I say it's tragic? Because when you get to the planned end of a study and you find that there's little evidence that the treatment works, it's very commonly the case that you could have stopped the study way earlier. So if something is not going to succeed, the earlier you fail, the better. And this is one of the biggest ways where Bayesian design has an impact, is stopping earlier for futility. Don't waste the money if you're not going to have much of a chance of treatment benefit. So in the traditional world, you need a final sample size to know when your alpha is spent. And the idea of spending alpha is at the root of a lot of the problem. Fixed budgeting, I already mentioned, but the calculations that we do are voodoo because they depend on an arbitrary alpha, beta, or 1 minus power. And then this delta, the effect to not miss, is typically gamed. It's a very distasteful game that takes place in government researchers, pharmaceutical companies all over. And you also need an accurate standard deviation to do a power calculation, or if it's a binary event, you need to have an accurate estimate of the event incidence. That is typically overestimated in power calculations. Whereas the physics approach just says, we don't know all this stuff. Why are we basing a design on things that we don't know? Why don't we just experiment until we have an answer that is helpful? If you're using a Bayesian sequential design, you don't even need a sample size calculation. Now, most people would want to know an average sample size so they could calculate an average budget. But there's nothing in the Bayesian approach that would make you calculate a sample size. So you can be very much like a physicist. With Bayes, study extension is trivial. It requires no alpha penalty whatsoever. So it's logical to recruit more patients if the result is promising but not definitive. Say the probability of benefits between 0.7 and 0.95. Analysis of cumulative data after new data are added merely supersedes the previous analyses. So when you've done analyses along the way, these are interim analyses. When you get to a new analysis, the previous results are merely obsolete and are totally forgotten. That's why there's no multiplicity issue in the Bayesian world. So is alpha a good thing to control in general? This is where the deepest arguments happen throughout the world in statistics. Alpha is often called a type 1 error, and it's really misleading and has caused a lot of harm to call alpha an error probability because it's nothing of the sort. So by thinking it's a type 1 error, it's actually making people very confused. Alpha is the probability of asserting something, and in a simple way of saying it's the probability the p-value is less than 0.05 if the null hypothesis is true and you're operating at a traditional 0.05 level. So alpha is nothing more than the probability of asserting something when any assertion is by definition wrong. Now, if you look at this formal definition of alpha, that's as far from an error probability as you could possibly get. And alpha increases as the number of looks go up. Alpha is not the probability of making an error in acting as if a treatment works. So we endow alpha with all sorts of magical properties, and that's at the heart of rigid design and wasted money, in my view. Alpha includes the probabilities of things that might have happened. So even if we have very strong evidence of efficacy at a given time, our design had the possibility of showing an efficacy signal at other times had the efficacy been exactly 0. This possibility is alpha. So what you're saying is you have current evidence that is all that matters for making the optimum decision. And all of a sudden, you're going to tamper that evidence by other opportunities you had to have other evidence. And from the rules of evidentiary assessment, that doesn't make any sense. And I think it's only in medicine that people tend to really, really embrace this. So Bayesian methods deal with what did happen and not with what might have happened. And that is a game changer. So asking one to compute alpha for a Bayesian design is like asking a poker player who wins more than $10 million a year to justify his ranking by how seldom he places bets in games he didn't win. So you can see that the way decisions are made, they're made forward in time. And a poker player has certain information available to him at a certain point in the game, uses that information to make a prediction of being able to win the pot, and places a bet accordingly. The poker player doesn't get ranked on other opportunities that he had to make bets or in bets in games that he lost. The judgment of the poker player's skill is based on the same way we should be judging a clinical trial as do we get the right answer? Do we make the right decision, whether it's placing a bet or whether we are acting as if the treatment worked? Does the treatment actually work? So controlling alpha leads to conservatism, whereas in a Bayesian sequential design, the expected sample size at the time of stopping will go down as the number of looks goes up. So you can take that to the limit and look at the data after every patient is enrolled and followed in the study. And if you do an analysis every day, you will get the lowest expected sample size. So what should we control? Just like in life, you want to control the correctness of the decisions that you make. This is what Bayes is all about. It's not about the chance of observing surprising data if you assume the treatment is ignorable. It's about the probability of being wrong in acting as if a treatment works. So if you calculate the Bayesian posterior probability that the treatment works, efficacy, 1 minus the probability that the treatment works is the probability that it doesn't work. And that is what I would call a decision error. And we want to control decision errors. We want to control how likely it is that if we act as if a drug works, that it really, in all truth, doesn't actually work. That is completely unrelated to alpha in the traditional statistical world. So the way you control the probability of a decision error is controlled by the prior distribution and the data and the statistical model, the outcome measure you choose, and your sample size, and probably some other things I haven't listed there. So for example, in the Bayesian world, you may say, we're going to look at time until death, and we're going to summarize that with a hazard ratio. We might calculate the probability that the hazard ratio is in the right direction. It's less than 1 for the new treatment, given the current data and given the prior distribution. And if that probability that the drug benefits mortality is 0.96, that means the probability that the hazard ratio is in the wrong direction is by definition 0.04. So a really great thing about forward probabilities in the Bayesian world is the probability of efficacy defines its own error probability, and it's a true error probability. This is the probability of making a mistake if you act as if the drug lowers mortality, and it doesn't actually do that. So this is exactly what an error probability is supposed to look like. Alpha has absolutely nothing to do with that. So the shocking truth of Bayes is that it controls the reliability of evidence at the decision point, not at the study planning point where you might conceive of what happened. So the way alpha is considered is it's a pre-study tendency for data to be extreme under an unknowable assumption that the treatment is ignorable, the treatment does nothing. So this link here, I've got a lot of simulation examples that show how safe it is to do completely continuous analysis in the Bayesian sense, and how it doesn't disturb the probability of making a mistake in any way, shape, or form. So by not having an alpha penalty, you're gaining power with a Bayesian design, and by being directional, there's no discussion about two-tailed versus one-tailed. There's no possibility of making a claim for an increase in mortality. We're only interested in probabilities about decreasing mortality. And so Bayesian assessments are directional, which gains a little bit of power. You allow for infinitely many data looks. And then if there is treatment effect modification, such as interaction, you're also able to borrow information. There's no solution to this problem in the frequentist world. For example, if you wanted to have different treatment assessments for males as for females, you would put a Bayesian prior on the treatment by sex interaction, and you would have a discussion ahead of time of how much you're going to assume the males are more similar to females in the treatment effect than they are different. So that gives you a very coherent way to borrow information from males to females when you're trying to bring evidence for the treatment effect that's sex specific. Now we turn to a different subject, which will apply whether you're doing traditional analysis or Bayesian analysis, and that is the outcome measure. This is one of my biggest frustrations as a statistician working in clinical trials, that we see a lot of very inefficient outcomes. So in non-rare diseases, why is it, say in very common cardiovascular diseases, you need between 6,000 and 10,000 patients for most of the pivotal phase 3 studies? Why is that required? And we don't require that in other settings. Well, it's required because you're using a low information outcome. Time to a binary event. A binary event is the smallest amount of information. That's 1 bit of information in terms of Shannon's information measure. So when you're collecting 1 bit of information, that's so low that you have to study a lot of people to have sufficient sensitivity, whether it's frequentist power or Bayesian power. And you're not breaking the ties. So having a binary event means you're not breaking ties. So you're counting a small myocardial infarction the same as a big myocardial infarction, and you're counting a small heart attack the same as a death. And if somebody has a non-fatal heart attack and goes on to die a month later, the death is completely ignored because the vast majority of cardiovascular trials use the time into the first event. So if the first event is a non-fatal MI, you die a month later, the death is completely swept under the rug. So the reason you need 6,000 to 10,000 patients is depending on your outcome incidence, that will generate about 462 events. And you need 462 events to be able to estimate a hazard ratio to within a fold change of 1.2 or multiplicative margin of error. Now, to contrast that with a continuous outcome measure, instead of needing 6,000 patients for a time to event analysis, you might need 384 patients to estimate a difference in means to within 0.2 standard deviations. And take that idea of high information to the limit where you can use each patient as her own control in a crossover design to detect that same sort of difference in a crossover study would require 96 patients total in a two-period crossover design. So as the event incidence goes down, censoring goes up, and also the time to event analysis becomes same as a binary outcome, ignoring time. So when you have a rare outcome, the time until the event doesn't help you statistically. It's just like a binary outcome. So we need to think about general outcome attributes to gain information, lower sample sizes, increase power. So we need to think about the timing and severity of outcomes. We need to handle terminal events such as death, non-terminal events such as heart attack and stroke, and recurrent events such as hospitalization. We need to break the ties. And there's a blog article linked here, which shows graphically how breaking the ties, the more ties you break in your outcome measure, the higher the power of the comparison of two treatments. The maximum power is going to occur when there's only one patient at each level of Y. That would be a continuous outcome like blood pressure with very few ties, if any, in the data. So that's a continuous outcome is your maximum information, maximum power, minimum sample size outcome until you get to longitudinal, which helps even further. So what is a fundamental outcome assessment for the vast majority of clinical trials that I've ever seen? You can think of it this central way. And once you think about data as longitudinal, it opens up a wide variety of powerful analyses, unlike doing time to first event analysis. So think about in a given time period, week or a day, what is the severity of the worst thing that happened to the patient? A way to judge the success of a treatment would be that it makes the worst thing that happens to a patient less bad than placebo does. So think about this as a kind of a fundamental piece of information that you should be analyzing. If you have expert consensus where you can show that on a given day, people are good at ranking the severity of the outcomes, we'll rank a hospitalization not as bad as a heart attack and maybe that not as bad as a stroke. You just need a consensus on which outcome is worse than which other outcome to be able to do this. The spacing of the outcome categories is completely irrelevant. So if you say death is worse than the other outcomes, the way that we're going to analyze that, it makes no assumption about how much worse it is. It's just worse. So this avoids having to define additive weights for multiple events in the same week. It's a hierarchical severity scoring. But you can grade events and you can code common co-occurring events as a worse event. So if somebody had both heart attack and a stroke, you could count that as its own category as worse than either one of those events separately. You can translate an ordinal longitudinal model to obtain a variety of estimates. So you think about your fundamental unit of measurement in a clinical study. That unit of measurement is fundamentally a longitudinal assessment of patient status. And once you think about patient status, it makes the analysis much easier, much more clinically interpretable. And you can interpret the analysis in a variety of ways, such as time until a condition happens, the expected time in state, like how often is somebody disabled, how long is somebody sick, how long is somebody in the hospital or dead. And you can calculate the probability of something bad or worse happening to the patient over time by treatment. These are all clinical readouts that are very, very clinically relevant. And a longitudinal model can give you all of these. So one of the biggest mistakes I see clinicians making when they're designing clinical trials is trying to reduce longitudinal patient status into a time to event analysis. The worst single example I've ever seen is in a renal damage study of having time until doubling of serum creatinine. that violates just about every statistical principle of design. So we have what's called a Bayesian partial proportional odds model. And this is the only method that I have ever seen that gives you a formal assessment of something I hear people talking about all the time, is what is the level of evidence that the treatment lowers mortality in the same way it improves non-fatal outcomes. So this way has a very formal differentiator to differentiate the treatment effect for different components of the outcome, and also to borrow information. So I think one of the most useful ways to borrow information is to say, we're going to assume there's some commonality that the way this treatment lowers mortality is going to spill over to the way it makes patients have better functional status. But we're not going to assume there's a one-to-one relationship between those two. So we're going to borrow information between the functional status outcome and the mortality outcome. You don't have to do that, but it's an option in a longitudinal model. And a Bayesian borrowing of information across different outcome components is something you probably never heard of, but this model really opens this up. So an ordinal model also elegantly handles partial information. At each day or week, the ordinal model can use an outcome or patient status that's left censored, right censored, or interval censored when the range of a scale was not measured on a given time period. So here's what the raw data would look like. This is a regular binary event setup where you're assessing patients dead or alive for 3 weeks. Here's a patient who was followed for 3 weeks and did not have a death observed, so they're censored at 3 weeks. In longitudinal data, that's a 000. Somebody who had a death in the second week, they're going to be a 01, and then their record terminates here. So that's an absorbing state or terminal event. And if you apply the longitudinal binary logistic model to this, the odds ratio you get from that model is almost exactly equal to the hazard ratio in a Cox proportional hazards model. But you can take this so much farther. So let's suppose you had at home hospitalized myocardial infarction or death. So somebody who is hospitalized at 3 weeks, they're rehospitalized at 7 weeks, they have a heart attack at 8 weeks, they stay in the hospital, and the follow-up ends at 10 weeks, would have a 001. They're at home these 3 days, and then they're hospitalized again. They have a heart attack. They're still in the hospital, still in the hospital. So this is what the longitudinal data looks like. Something funny happens when you start analyzing raw data. As you get closer to having maximum power, and you also get a much easier clinical interpretation. If you had quality of life as part of your outcome scale, where 0 to 6 is your quality of life, where poor is 6, and 7 is a heart attack, 8 is a stroke, 9 is death, you might have somebody starting out at really good quality of life. You don't have the quality of life measured on day 3 or week 3, but you know they didn't have an event. So that would be interval censored. And the statistical method handles that elegantly with no imputation. So we just know that the patient status on week 3 is somewhere between 0 and 6, and that's exactly what goes into the analysis. And then we have quality of life 3, 3, 4, and 5, and then we have events. Here's somebody that, in addition to not having the quality of life assessed in the 3rd week, we know they didn't have a stroke or die, so they're not an 8 or 9, but we don't know whether or not they had a heart attack. So their data in that week would be either a 5 or a 7. If they had a heart attack, it's going to be a 7. If they didn't, it's going to be a quality of life 5. So you can see how you're using partial information, and you can put a continuous variable in this scale for the first 200 levels. For example, I might have serum creatinine as a scale, and if the patient needs emergency dialysis, that would be 1 level worse than any serum creatinine that you could observe. So this statistical model that would be one way to analyze this is a proportional odds model with covariate adjustment. It handles intra-patient correlation with a very simple Markov process. You can do it other ways. This extends a binary logistic model, and it's a complete generalization of the Wilcoxon 2-sample test. And so if you like the Wilcoxon test for judging which group of patients is doing better, you have to like this method, which gives you exactly the Wilcoxon test as a special case. It doesn't assume anything about the Y distribution, and it doesn't use the numeric codes of the Y. It just uses the ordering. So one of the outputs you get from this is a treatment to control odds ratio. You can convert that to the Wilcoxon statistic, which is the probability somebody on B does better than somebody on placebo. This is called a concordance probability C index or probability index. And if you calculate the odds ratio from a proportional odds model and use this formula, you get virtually exactly the Wilcoxon statistic. So there's a one-to-one correspondence between the Wilcoxon statistic and this method without assuming proportional odds in any way. You can calculate the probability of a certain outcome or worse as a function of time or treatment. You can estimate the time gained in good states, like the difference in mean weeks unwell. And you can compute posterior probabilities that the treatment affects death differently than it affects quality of life. That's a very formal part of an ordinal model using what's called the partial proportional odds model. Brings this home a little bit. This is one way to show the results, is you have here follow-up time in weeks, and then we have the probability of an outcome if you're on treatment B and if you're on treatment A. So treatment A is having worse results after randomization because the probability of being hospitalized or worse is always higher for treatment A. Let's say so treatment A is doing worse than treatment B. But you could say we're going to calculate the probability of myocardial infarction or death. That would be what we want to estimate. The same model estimates that. And so hospitalized or worst here means hospitalized, heart attack, or death. This is just the top two levels. This is the top level. So this is the probability of being dead as a function of time and treatment. So one model gives you all these estimates, and I think it's a very clinical way to look at it. This is some examples of how power is gained by looking at the data this way. This is a reanalysis of a study called Violet, which is a study of vitamin D therapy for patients with acute respiratory distress syndrome. So these are patients in the intensive care unit, randomized to placebo or high-dose vitamin D, and the primary endpoint is mortality, where there was evidence for a slight increase for mortality due to vitamin D. You know, we've seen as many or more studies where vitamin D hurts you than it helps you. But there was an ordinal outcome collected at each day for 28 consecutive days. And this is a really good way to visualize that. This is the first 14 days of follow-up. We have had clinicians really liking this sort of stacked bar chart representation when your events that you're getting the patient status for is fairly discrete, although this applies to continuous outcomes also. So the status we have being at home, being in hospital or like a nursing facility, being on a ventilator or having a acute respiratory distress syndrome or being dead. So the death is an absorbing state. So the incidence of death is always increasing. It can never go down. But that's not true of these others. So some of these you'll see go up and down. This height of this bar here is the probability of being on a ventilator or having ARDS as a function of time and treatment. This is the probability of being in the hospital. But if you stack all these up and look at this height compared to this reference here, this is the probability of anything bad happening to you as a function of time. And this height here from the top is the probability of being in the best outcome state, which is being at home. So this is what it looks like to summarize longitudinal ordinal data. And I think it's very clinically relevant and very interpretable. Now, in one sub-study I did of the reanalysis of this study with 28-day follow-up is I could estimate the relative efficiency of having follow-up at more than 1 day versus the efficiency of having the follow-up at only 1 day. So let's suppose in a 28-day follow-up, you followed people just at day 14. How much more information is it to have them at day 7, 14, and 21, or to have them all the way measured their status on 28 days? Even with a high correlation within patient, I calculated that every 6 days of follow-up has the same statistical information as enrolling a new patient in the study. So you think of 6 measurements on one patient contributes the same power as enrolling a whole new patient when you compare that to assessing the patient outcome in only a single day. Also, when you look at the other methods of analysis, such as time to recovery analysis, time to recovery is statistically a horrendous outcome, has terrible properties, and it also loses power. So the longitudinal ordinal model in the power simulation had this power. A time to recovery analysis with a Cox model and competing risk analysis had this power. And if you looked at ventilator-free days with death coded as a -1, which is a very common and very defective way to do critical care medicine research, it really loses a lot of power. So you can see that using the raw data gives you this sort of instant benefit. Raw data means higher information. So the principles for choosing outcome measures are increasing power by breaking ties, getting close to the raw data. You are actually more relevant to what patients are experiencing. You're respecting the timing and severity of outcomes, and you can do automatic risk-benefit trade-offs by including safety events in an ordinal outcome if you choose to do so. I'm going to very briefly cover this topic. This is something where you get something truly for free. And this is still poorly understood, that covariate adjustment is a way to gain power no matter what your outcome variable is. And whether you're using Bayesian or frequentist, it doesn't matter. So covariate adjustment has almost nothing to do with baseline balance in a randomized study. It has to do with outcome heterogeneity. It is stunning to me, and I'm almost pass out when I see a study that has patients who are between 20 and 90 years old, and the analysis doesn't recognize that older patients die sooner than younger patients. This is almost a criminal statistical analysis to not recognize this. So having a variety of ages in your study and not adjusting for ages at covariate means your outcome heterogeneity within a treatment group is massive. It's the same way if you have a 6-minute walk test and you don't adjust for the pre-treatment 6-minute walk test, you will lose a lot of statistical power, and you'll have to randomize a lot more patients. So I'm going to gloss over some of these other aspects, but there are examples where using a covariate can cut the sample size in half. So let's say if you had a continuous outcome and the covariate has an R-squared of 0.5 against the outcome, that means not using the covariate, you need to double the sample size as compared to using the sample size. So we're not talking about trivial effects here. And covariate adjustment is free. You know, we have every clinical trial measure some well thought out baseline measures, including age and disease severity. And using that in the analysis is just free, doesn't cost a thing. And we're still seeing it very uncommonly done. This is one thing FDA invites sponsors to always consider is covariate adjustment, and has a wonderful guidance document on that. Computing change from baseline is one of the biggest statistical sins that I see in my practice. Computing change from baseline makes assumptions that just are not true, and it assumes you don't have floor and ceiling effects. So I see people getting wrong answers from analyzing change of baseline, which has massive assumptions. You have to have a linear relationship between pre and post, and the slope of that relationship needs to be 1. And so it's really even inconsistent with a parallel group design. So baseline needs to be treated as baseline. It needs to be a covariate. And I've got a lot of teaching materials about that. So the take-home messages are, don't take sample size seriously. consider sequential designs with unlimited data looks to maximize the Bayesian power and minimize the sample size and to quit wasting so much money. Alpha is not a relevant quantity to control or spin because it's completely unrelated to decision errors. Choose a high resolution, high information outcome variable that will have a huge effect on the power. Longitudinal ordinal way is a general and flexible way to capture severity and timing of outcomes. Always adjust for strong prognostic outcomes and don't stratify by treatment in table 1. Because if you stratify by treatment in table 1, you don't really understand randomization. So there's some references here, and I'll just, it goes into a lot more detail. This is my newest stuff. This is a detailed Bayesian clinical trial design and simulation of how it works when you have continuous looks at the data. And it covers a lot more things. So I'll stop there. I look forward to some discussion and hope you will consider some of the things I propose after suffering with the way clinical trials are traditionally done for many decades. Thanks for having me.
Thank you so much, Frank. Really excellent. There was a lot of data presented and a lot of brilliant examples. So just to tell to our audience that the link to the slides will be available on our website and the recording will be on YouTube tomorrow. So please, I'd like to see the show of hands for you to just ask questions. And I have a lot of questions for you, Frank, but Nicholas Irons, please go ahead.
Hi, can you hear me?
Yes.
Great talk, Frank. That lots to chew on there.
Thank
yeah.
you.
I guess a few questions, or well, one comment is I really liked how you called sample size calculations voodoo, because that's one thing that really makes me feel gross whenever I'm, I've done some statistical consulting work with people, and I'm like, oh, this is very hand wavy. So I appreciate that. I guess I'm curious about your thoughts on the use of like Bayes factors for inference as opposed, because you were saying, let's just look at a posterior probability of efficacy or something like that. And that's when we determine that we've, come to the right conclusion. But when you're explicitly comparing a null to an alternative, you can get different results than you would if you're just using the alternative model and looking at a posterior probability there. So I'm curious about your thoughts on the use of Bayes factors in clinical trials, especially sequential clinical trials. And then I guess I also had a question, and just feel free to answer one of these and not spend too much time. I don't want to take up all the time, but say you like categorize your continuous outcome into 200 categories, as you said, ordinally. Does that mean you have to estimate like 199 parameters there, or yeah, how does that work? So anyway,
let me
thank you.
start with that one. These are both wonderful questions, Nicholas. The idea when you have a continuous variable is not to categorize it at all. So let's say you had 10,000 patients in a study and you had 6,000 distinct values. You would have 5,999 intercepts in the model. And we have efficient R software that will handle that case very efficiently. So there's really, once you get past 6,000 intercepts in a model, it gets to be slow. But there's really no categorization needed in the vast majority of outcomes to use an ordinal model. That's why I don't use linear models anymore. I always use a semi-parametric ordinal model, so I don't assume normality and things like that. So the intercepts are really easy to handle. You just need to use some computational tricks. They're forced to be in order. That's why they behave so well, even though there's so many of them. The interesting thought about Bayes factor, the reason I don't use Bayes factors, there's two reasons. They invite you to think about point hypotheses, and I am never interested in a point hypothesis. I'm interested in evidence about intervals. So I want to know, I may have a similarity interval, and I want to calculate a posterior probability that the treatment effect is within a similarity interval. So it's just so easy to do that with posterior inference. But the bigger reason even is the Bayes factor idea doesn't properly generalize. So I've got a section in my Bayesian course about dealing with multiple outcomes where you might want to calculate the probability that you reduced mortality by any amount, or you reduced blood pressure by at least 5 millimeters of mercury. So that would be a compound statement. You're assessing evidence for either mortality or any mortality reduction or a big blood pressure reduction. I don't think Bayes factors would handle that. That's just a compound assertion in a posterior probability. So I just see posterior probabilities as covering all the general cases that I need.
Okay, thanks. I would love to talk to you more about this. Can I email you? Is that okay?
Oh, yeah, yeah.
Okay, great.
We also have a lot of discussion boards. I mentioned at the top here, datamethod.org is a great place to keep discussions going related to things I talked about. Many of the things already have topics created in data methods, but you can create new topics there.
Awesome. Thank you. Great talk.
Sure, thank you.
Thank you, Frank. Could you please stop sharing the screen, and this way we'll see the audience and those who raise their hands. We have 2 questions in the chat, so I'll read one of them and then I'll go to David to ask his question. So the question from the chat is from Marianna Nodale, and the question is regarding the longitudinal odds model. How do you assess whether your model is misspecified? More detailed on the question, if you can see that, Frank. So
yeah,
go ahead.
that's one of the first questions that comes up when I give talks on some of this material. And I've devoted like 5 different blog articles to answering that question, and I'll try to give a brief answer. But one of the interesting things about that is people tend to worry about the proportional odds assumption more than they worry about anything in their career. I've never seen anything like it, and I don't understand it. The proportional odds model was invented in 1967, and we know a lot about it by now. And I see people questioning it when, if you gave them a traditional repeated measures mixed effects model, they don't check any of the assumptions of that. And that makes far more assumptions than the proportional odds model, not even close. So there's a little bit of a disconnect in practice where people don't question assumptions of things that are highly parametric because they've seen them multiple times in their practice, whereas they do question other things. But getting more to the point, the proportional odds model, it has the Wilcoxon test as a special case, and I have multiple blog articles showing that. So if you like the Wilcoxon test to tell you which treatment is better, you have to like the proportional odds model. But to be specific about that, what goes wrong with the proportional odds model is when you have different, you have this non-parallelism and you have these different odds ratios at each level, as Marianna mentioned in the chat, is it will cause you to get incorrect predictions at individual outcome levels. So if you wanted to get a mortality prediction or a probability of having a heart attack and not dying, something specific, you can get a wrong specific probability estimate for at least one of the outcome categories if this proportional odds assumption is not satisfied. But it doesn't affect in any way, shape, or form the assessment of which treatment is better. So the proportional odds model, it agrees 100% of the time with the Wilcoxon test in judging which treatment is better. So the Wilcoxon test statistic comes out to its null value if and only if the computed odds ratio is exactly 1.0. And the odds ratio always gives the same direction of a treatment effect as the Wilcoxon test, no matter if the proportional odds assumption is violated to an incredible extreme.
Thank you, Frank. That was very clear. So David, please go ahead with your question.
Yes, I just love your way of explaining the difference between p-value and the posterior probability. The probability that a positive result turns out to be nothing is clearly what you want. The wording, as you say, is quite difficult to take in. I'm wondering to what extent that's the same as the distinction I like to make between equal to or less than. The point is that when you do it in well, it applies to a likelihood. It's not necessarily Bayesian, but why consider results that haven't been observed? If you're comparing two hypotheses, you can compare the say the probability that the p-value is equal to. I'm sorry, I'm not explaining this very well. I'm getting too old, but I hope you know what I mean.
And I think, I don't know if you're alluding to the fact that a p-value, it relates to things you didn't observe, and it calculates the probability of data more extreme than what you observed. It doesn't even use the probability of data as extreme of what you observe. So that inequality thing is maybe what you're getting at is very fascinating and very misunderstood. But I know you've spent a career trying to explain these things and have fought the really good fight. So I'm always glad when I see you involved in these discussions, David.
Thank you very much. It's entirely a post-retirement hobby for me, actually. Spent most of my life on stochastic processes of single molecules.
Wow, cool.
Thank you so much. So we have quite an avalanche of questions. So let's see how far we progress. We can overrun a little bit. So I'll give a word to Robert now, who's holding his hand. Please go ahead, Robert.
Hi, Frank. Thanks again. Wonderful talk, as always.
Yeah.
I'm slowly getting deep enough into this to start thinking about how it relates to some of my work. And one of it is the tools I use. One of those is the time-dependent covariates in traditional survival methods. How does I mean I love the proportional odds, ordinal, longitudinal. Can you fit time-dependent covariate in there? And because I know you often talk about the time-dependent covariate, the intercurrent events that we are now starting to call them, that those are part of the outcome. And if that's what you're measuring, then that's what you're measuring. But a lot of trials, there's those sort of supplementary analyses trying to understand mechanism and why did the disease progress in this way, where I think some of those intercurrent events become more important to be analyzed as a distinct thing. So I just wonder if there's a place for that, or is that outside of the space of we have an efficacy question, let's answer it, darn it.
Yeah, I think it's a wonderful question, and it relates to supplementary analyses that don't really respect the intention to treat design, but are still very interesting as secondary analyses. That's when you use updated covariates such as advancing disease stage, and you might do some kind of mediation analysis related to that. It may not be your primary analysis, which might only adjust for disease stage at baseline. But I think the longitudinal ordinal model, it admits time-dependent covariates very easily. And it's kind of in the sense that certain econometric models do this, even with ordinary linear models with time-dependent covariates. You mentioned originally the proportional hazards model, and one of the things Bayes does is very unique, that is solving a lot of problems. If anybody would pay attention to it, people are worried about proportional hazards just like they're worried about the common odds ratio. And so in the partial proportional odds model, it's exactly the analogy to the time-dependent covariate in the Cox proportional hazards model, which is not an updated covariate. It's a time-varying baseline variable effect. So it's just a treatment effect. Bayes makes this actually coherent, and it's not coherent without Bayes. Why do I say that? If you allow the treatment to be time-dependent, you're fitting a whole degree of freedom for that, and you can dilute the power of the treatment effect. If you have a Bayesian prior on the amount of crossing of survival curves or the amount of non-constancy of the hazard ratio, you put a prior on that, and that prior says we're going to borrow information across time. And as the sample size grows, we're going to allow the hazard ratio to depart from being constant because the prior will wear off. And so ultimately, if you have a crossing of survival curves, the model will estimate that. And when the sample size is very small, it borrows information across time. The The traditional solution to these problems is to devote parameters, and people don't want to do that because it can dilute power. The Bayesian is the only coherent solution to this because it allows you to devote partial degrees of freedom to things that you hope you don't need, like non-proportional odds and non-proportional hazards. It's just like in an interaction analysis, you have a prior probability on the treatment effect being similar for males and females. It's in the same exact spirit as that.
Yeah.
So your question touches on a lot of different interesting things.
Thanks, Frank. I'm glad I'm not barking up the wrong tree here.
Not in any way.
Thank you, Frank. So, and with my next question, I want to combine 3 questions, mine and 2 from the chat, and then I'll give a word to Youngsji, who's holding her hand. So, my question to you was, where are we if we look at, I don't know if you've done statistical analysis on that, but frequentist camp versus Bayesian camp in terms of top thinkers or implement those people who actually implement trials, where are we now and how is this evolving, this whole dialogue? And the 2 related questions from the chat, one from Francois is, are the major current limitations of clinical studies are inherent due to regulations from responsibility from FDA and EMA, them expecting particular sample size, expecting particular power and alpha errors? And the follow-up question, somewhat within the same topic, is from Phoebe, who is representing the funders, and she's saying with funders getting more applications with Bayesian design, and we're not equipped with knowledge of how to evaluate these and how to deal with that. So I guess to summarize it all up, the field is changing. How is it changing? And how regulators and funders are reacting to that, and what should happen, or any suggestions?
These are hot topics, Lisa. So I'll just take a stab at some of them. One of the things that's evolving the fastest is people are getting impatient with p-values, especially large p-values, when the confidence interval is medium wide, but the point estimate for the treatment effect is huge. So we're starting to see more and more respected medical journals, people saying that the original interpretation was wrong. The original interpretation when the p-value was 0.1 was that the treatment doesn't work. And that means you don't understand a p-value. That has nothing to do with a p-value. So p-value 0.1 means you haven't brought evidence to reject the supposition that the treatment doesn't work. And that's all it is. And so if the treatment lowers mortality by 20% and the p-value is 0.07, we're seeing a lot of papers where people are doing Bayesian reanalysis to say how it should have been done in the first place, and we have a 0.97 probability that the treatment lowers mortality. Would you use that drug, or would you have your mother take that drug if it has a 97% chance of lowering mortality? So you're seeing these a lot of these reanalyses come out with a lot of people being impatient with these p-values, especially at the margin. A place where things are evolving very nicely is when there's really logical borrowing of information, and where the FDA is making huge progress is in applying the results of adult clinical trials to children. And so they're using a lot of wonderful Bayesian methods for saying how applicable are the adult data to the kids, and we'll put the probability of applicability of the adults as part of the prior for the kids. So you're also seeing work done in the rare disease area. FDA has a huge number of initiatives going on, but there is a misunderstanding that there are regulations that prevent new things from being used, and there's no regulations about that. There's nothing encoded in a regulation that says you have to use a p-value. The regulations are about having substantial evidence, and sometimes you have to have 2 trials showing substantial evidence. The actual approach for showing evidence is not codified in any law anywhere that I know of, but it is codified in tradition. So the biggest challenge to people that want to do new things is fighting tradition. And the biggest fight of tradition is, if you look at how people are educated in graduate school, very few people are exposed to Bayesian methods. including statisticians. And there are very famous graduate programs in the US where the vast majority of the students going through those graduate programs, unlike our Vanderbilt program, which is multi-denominational, they go through these other graduate programs without any Bayesian understanding whatsoever. So that is actually holding us back, because that means there's not enough reviewers that understand it. Now, speaking of funders, the most conservative people that I've ever worked with are NIH. So in terms of being a biggest funder in the US, there's a lot of tradition and a lot of good tradition and a lot of rigor to clinical trials, a lot of good rigor. But the adherence to alpha and p-values is pretty daunting. And it's very slow to change. It's slower to change at NIH than it is in pharma companies and FDA, in my experience.
Very interesting. Lots of work to be done. So, Youngsi, please go ahead with your question.
Okay, thank you, Frank. Great talk. I work from a very practical background, so I'm going to bother you with a bunch of practical questions. So the first thing is I collaborate a lot with neurologists, and they have a bunch of like ordinal neurofunctional outcomes like modified ranking scale in stroke. And I often see like in neurological trials, it's become a routine that they first test for proportionality, and if it is not violated, then proceed with proportional odds model. If it is violated, then they do some Mann-Whitney test and report some win odds or probability of treatment A being superior to treatment B, kind of effect size measure that closely associated with Mann-Whitney test. But I think currently my work is focused on more like fundamental things of why people are so worried about proportional odds assumption. I don't think there is like, first, of course, is like they have so much focus on p-values, but as you said, the proportional odds model and the Mann-Whitney test, they basically give the same p-values. I think more if you ask neurologists why they are so opposing this non-proportionality, is that they feel if you see this divergent treatment effect, then it's probably not okay to summarize with a single number, whether it's a common odds ratio or win odds from the Mann-Whitney test. So I'm like, maybe our argument around our debate around proportional odds assumption is really about the interpretability of the treatment effect estimate instead of p-value or power, because in this case, we might argue that a single summary measure can be misleading, and maybe in this case, it's better just to present people with all the proportions of different categories and describe like without any statistical models. And my second point is like we feel like with ordinal outcomes, sometimes it's not really capture all the information from neurologists, because of course, ordinal outcomes is way better than a dichotomy outcome because you have more like information. But sometimes if you ask a neurologist to like, they would feel like, okay, a transition from score 3 to score 2 means much more than a transition from score 2 to score 1, and but this kind of information is not contained in ordinal outcomes, because ordinal outcomes, they only rank categories. So we're thinking, okay, maybe a possible solution is to actually attach utilities or any kind of numbers to these ordinal outcomes and make them numerical, such that we can reflect this varied clinical importance of these categories.
Let me start with that last one and work my way back. So the utilities is a really important topic, and it's very seldom taken seriously, and it needs to be. One of the interesting things is if you really do have utilities, you have to analyze them with an ordinal model, because the distribution of utilities doesn't fit any standard parametric model. So you're already having to use a proportional odds model when your outcome is continuous utilities, because there's floor and ceiling effects and bimodality and other weird things that go on. The thing about the ordinal models, we can't usually get utilities. It takes careful time trade-off studies and so on. I think you can show that the ordinal model is a good approximation to the full utility analysis. It may not give quite enough credit for lowering mortality, but it's going to give, it's going to count mortality as worse than anything else. So it doesn't use anything about the spacing, but there may be certain ways you think it under rewards lowering mortality, but in general, it doesn't under reward. In general, it does reward that. So, but I think it approximates utility analysis. The other things you mentioned, I'm very interested in the Rankin scale because it is ideal for a proportional odds model. And we wrote a paper related to that. But I see people analyzing Rankin scale dichotomized, which is criminal. I mean,
Yeah,
that
indeed.
should get jail time in my view, because that's an ordinal variable. It was derived to be ordinal, and you have to treat it as ordinal. Otherwise, you're just going to have a bigger sample size that has a penalty for idiocy. I hate to say something that strong, but you don't want to just throw information under the rug. The thing about testing proportional odds is that test was implemented in SAS in about 1991. That test is invalid, and we pointed it out to the author of the SAS procedure in 1991, who refused to take it out of the procedure. I have just a real trivial example that shows how the p-values are totally invalid. And so people are trusting that procedure, and they should not be even using that procedure. I have a blog article that shows how to get a better test of proportional odds using the likelihood ratio test, which doesn't misbehave as much as the score test that SAS uses. But that SAS test, now we're past 30 years that should have been removed from SAS, and they were amply warned in the early 90s. I don't understand this. But one of my blog articles points out how testing assumptions is not really a great idea. What you want to find out is whether you don't make the assumption, do you get a worse result than if you make the assumption? And I have examples where, like, if you use different cut points and you mention giving all the individual proportions, that is almost always a worse result. And it's too influenced by noise. And so, and then you're going to lower the power a lot. So the idea of using different cut points is almost always worse than using the proportional odds assumption. But the other way to look at it is if you allowed things to be multinomial instead of ordinal, not only can you do a likelihood ratio test for whether you need to do that, you can do something more important, which is AIC. So if you calculated AIC for the model that assumed proportional odds and the model that didn't, it will tell you which of those models is going to predict the future better. And it's very commonly the case that if you don't assume proportional odds, you're going to get worse predictions, even with non-proportional odds, than you do by assuming proportional odds. So we need to move away from the classic goodness of fit that we're taught in school into measuring the impact of the choices we make. It's all about the impact, not whether some assumption is held or not. And I do want to mention one other thing while I'm on this high horse. The proportional odds assumption is exactly analogous to the equal variance assumption in a t-test. It's an exact analogy. And I see people going ballistic about the proportional odds assumption that do not assess the equal variance assumption in a t-test. That is not honest. That is just not being honest.
Indeed. Thank you. Great explanation.
Thank you.
Thank you, Frank. So let's go with the last question from Nicholas. And yes, there's one more.
Yeah, sorry to
Nicholas with
keep
the
you longer.
beautiful blue sky there.
Yeah, I live in Seattle, and this is the first
Nice.
sunny day in a while, so I figured I'd
All
take
right.
advantage of it.
Enjoy it.
Yeah, a few things. So you didn't really talk about adaptive designs. I'm wondering if you see, to me, it seems like kind of the best we can do if we're stuck in this fixed budget paradigm of at least trying to allocate our resources a bit more intelligently than just having a fixed design from the outset. I was also interested that you use the C index, because the C index is shown to not be a proper scoring rule for longitudinal models, which I don't know, you might be familiar with that. So anyway, so I'm curious on your thoughts of the C index versus the AUC, and then yeah, other things I'll bring up on your blog. The data methods website looks cool. I've been checking it out.
Yeah, just one comment on the C index is that it's not a gold standard, and so I like to use gold standards whenever I can, and the gold standard is always the log likelihood. So we need to use measures, and I've been doing a lot of work with generalized adjusted R-squared measures that apply to longitudinal and other data. And so those use the log likelihood, and those are going to be more sensitive, and they also work for sensor data and interval censoring and, you know, all kind of other situations. I do want to pick up on your comment about adaptive. I think the most underutilized thing is sequential designs, and sequential designs are a lot simpler than adaptive designs. They're not having adaptation, where adaptive designs give you additional information is when you're doing like dose adjustment and you've got more than 2 doses. Adaptive trials need there to be more than 2 alternatives before you can really do adaptation in a meaningful way. So dose finding adaptation can be really good, and there's some biomarker driven studies. I think what we learned from these studies, the uncertainty intervals at the end of the adaptive studies are wider than people wanted. And often we don't know quite as much as we thought we did because the adaptations didn't control certain cell sizes for sample sizes and so on. But they can still be super valuable. And I don't want to discount using like response adaptive randomization and all the other things. But I think sequential trials are so easy. And they also allow you to drop treatment arms at your whim, and, you know, with no adjustment for anything. So I would like to see the simple things used more often.
Thank you, Frank.
I
So
appreciate that.
yes, now last question from Alan, please go ahead.
Yes, thanks. Hi, Frank. I can't offer
Hey.
you a nice blue sky in the background, but a question on covariate adjustment we haven't touched upon. So I guess you were talking of traditional covariate adjustment for conditional estimates, but what do you think of covariate adjustment for average marginal estimates? There's also now, I mean, there's quite some guidance out there, and what can you specify what you were touching upon with covariate adjustment exactly?
Yeah, Stephen Sen and I wrote a very long blog article on fharrell.com showing severe problems with marginal estimates. And I've got a new editorial be coming out in the journal Clinical Trials in response to a new paper coming out there that points out a key mistake, which is the marginal estimates assume the clinical trial is a random sample from a population, and that's never true. So the marginal estimates are actually very biased. So you don't want to go to a lot of trouble to create an estimate that's biased when you don't know the probability weights to get from your sampling scheme in the population, because in a clinical trial, you don't have those probability weights. They're not known to you because people volunteer. They're never grabbed off the streets to enroll in a clinical trial. So the marginal estimates, I have a blog article you might be interested in with a title, something like marginal estimates are conditional. So you can show that the marginal estimates, what they're estimating depends on a covariate mix that's out of your control. And that makes them not transportable to the population. So people are going to a lot of trouble to get these marginal estimates that are not patient specific. They apply to group decision making and not individual clinical decision making. They're harder to compute, and they result in bias. So to me, it's a lose situation. But there's an industry that I'm going to anger probably 1,000 people today by saying this industry is a bit out of control, and people are loving it because it's new, but it is not really solving problems that clinicians ask anybody to solve.
Maybe epidemiologists, right? They like it because it's population level
and
random effects meta-analysis who try
I keep
to
asking
say this.
epidemiologists to point out a group decision-making problem for which a marginal estimate would apply, and I haven't gotten any takers. So even in epidemiology, I'm having a problem between conditional and marginal estimates.
Thanks. I have to read your blog, definitely.
Thank you so much, Frank. We have to wrap up, even though much of the audience, I think, would like to stay until tomorrow, keep asking you questions. So one thing is, I would be very excited if you came back with another talk that you proposed for Consilium and talk us through rare diseases and statistical issues in those clinical trials, massive on the agenda these days. But thank you so much for amazing learning, and for many it's uncovering a huge box of further things that we all need to explore through your writings and discussion boards and all the resources that you've shared. And thank you so much for our dedicated audience for staying and for your questions.
And the
So-
questions from the audience were absolutely phenomenal. So I love that, where they were coming from, and I love the engagement, and just thanks for having me, Lisa.
Thank you so much. So goodbye, everyone. And the recordings will be on our website and on YouTube tomorrow. Bye-bye.